biologist. Today in lecture we're going to talk about meiosis, which is how plants and humans and other organisms make their cells that they use for sex. Here is my beautiful flower. It's an amaryllis that I showed you in our class meeting. So beautiful. What's really exciting is that this plant is also making sex cells. So it's using meiosis to produce sperm and egg. This little structure here, see that little white thing? That's called the stigma. And down below it, in the base of my flower, so down here, is where all the eggs are. So I've got to get the sperm from a male plant, which this is both male and female, it does both. And I have to get it up here where it's going to grow a little tube all the way down to the eggs and will fertilize them. The sperm made by this plant is produced on the tip of these little things. Do you see there's 6 of them? Not the big fat one, that's the lady part. Over here, those things, those are called anthers, and at the tip is where they produce their sperm, which we call pollen. So you can see all that yellow pollen there. I took off the pollen sacs because it makes a mess in my kitchen and I don't like messes. But you can see the ruminants, all of that yellow is pollen, which is plant sperm. So that pollen, getting it on my finger, would get stuck on the stigma. I just fertilized my plant. And now the little pollen tube can grow down and fertilize these eggs in the base of my flower. Then, if the fertilization goes well, my flower will create a seed, which is essentially a baby plant ready to start growing into a whole new individual. So, super exciting plant sex. Now let's learn how my plant, or how a cat or a bird or something else, produces sperm and egg. We're going to do some notes, so get a pen and a piece of paper. Okay, let's start by reminding ourselves about the process of mitosis. So I'm going to draw the cell cycle. To do that, I'm going to draw a circle, and then I'm going to put a little arrow so I know it's a cycle. The whole thing happens again and again. And this is mitosis. You learned about mitosis last term. In mitosis, most of the process, so from here to here, this whole big part of the process is called interphase. So, all the way up to here from our starting point, 90% of your cell's life is just an interphase where it's doing cell things and it's just working like it should. And then if your cell needs to divide, like if it's a liver cell, you need to replace something, or if it's a skin cell, then you'll do mitosis. Mitosis is divided into a couple different steps. We had prophase, prometaphase, metaphase, anaphase, telophase, and then the last thing that happens, and it's kind of its own, it's not really part of the cell cycle, would be cytokinesis. If all of these words sound really unfamiliar, please review mitosis in your textbook. So, the whole goal of mitosis is to make 2 new identical cells. And we know that from last term. All of these cells are going to be diploid. And they're going to be somatic. Somatic means they're body cells. So everything in your body, your skin, your hair, your muscle, your neurons, your fat, all that cool stuff, everything except sperm and egg. That's what a somatic cell is. Everything except our sex cells. We're not going to go through the process of prometaphase, metaphase, and anaphase and telophase because you remember that from last term. But what we do want to talk about this time is what happens during interphase. We've got the first step of interphase, which is G1, then we have the S phase, and then we have G2. So we know that from last term. I want to talk about S phase for a second. What happens in S phase? Pop quiz. If you said in S phase you duplicate your DNA, you are correct. That's the whole goal of S phase of interphase, is to duplicate the DNA. I think S for synthesis, we're synthesizing more DNA. And I want to take a moment to draw that out. You have a whole bunch of DNA wrapped around histone proteins that become visible during prophase, and it looks like this. This is a chromosome. It's got this pinched in bit right here. We call that the centromere. And then on it, in the centromere, there's a series of proteins, and we call those proteins together the kinetochore. It's spelled Kinetochore, but pronounced Kinetochore. This gives us a little indicator of what's going to happen. Kinetic means movement. So your kinetochore is going to be used in moving your chromosomes around. And the kinetochore is where the spindle fibers are going to attach during prophase and prometaphase. So when I pull my chromosomes around to line up in the center of the cell during metaphase, I'm using all those proteins at the kinetochore to move the chromosome around. So that's where the kinetic part comes from. When I have two sets of DNA, because I've duplicated my DNA in S phase, they hang out together. So here's one chromosome. Here's the kinetochore. I've got another set of the same information, so it's identical to the first chromosome. Now I have two identical chromosomes. And we give these a special vocab word. It's kind of cute. What is it? It's sister chromatids. And they're held together at the centromere. The microtubules will attach to the kinetochores on each side, and during anaphase, we'll pull those two sister chromatids apart so that when we make our cell do cytokinesis, there's only one of each chromosome in your new cells. So that's mitosis. This is important because we're going to talk about the same vocab, except it's going to work a little bit differently in meiosis. So let's look at meiosis now. Let's draw ourselves a map, just like we did for mitosis. We're going to have the interphase occur, so this is going to be interphase. Then we're going to have Meiosis One and Meiosis Two. Oh yeah, that's a perfect circle right there, huh? So this is meiosis. The first part is interphase. Notice that interphase only occurs once. Then we have Meiosis One. And we have Meiosis Two. Lovely. There is not a second interphase between these two, so it only occurs once. Meiosis One is still broken up into those three steps, G1, S, and G2. We're going to grow in both the G phases. In the S phase, we're going to synthesize new DNA, so we're going to duplicate our DNA. And then Meiosis One is really where all the exciting things happen. Meiosis One is divided up into prophase 1, and you have to say 1 because Meiosis 2 is also going to have a prophase, and they're different. They do different things. Well, different things occur in them. So, prophase 1, prometaphase 1, metaphase 1, anaphase 1, and telophase 1. Then, after Meiosis 1, we're going to have cytokinesis. So that occurs in here. Then in meiosis 2, we're going to have prophase 2, prometaphase 2, metaphase 2, anaphase 2, and telophase 2, followed by, here, our second cytokinesis. To help us remember that it happens twice, let's call it cytokinesis 2 and cytokinesis once. So we're going to do cytokinesis twice. The reason that we're doing it twice is because we're going to end with 4 cells instead of mitosis, where you do it once and you end with 2 cells. So let's talk about the really cool stuff that happens in meiosis that makes this an amazing process. The first thing that's going to happen that's really cool is in prophase 1. So in prophase 1, I'm going to draw it over here, we're going to create something called the Synaptonemal Complex. And the way it works is this. I've got this chromosome. Let's say it's chromosome number 1. I've got 23 types of chromosomes, right? 1 through 23. This is chromosome number 1. Here's my kinetochore. There's the pinched in bit that shows the centromere. And I've duplicated it during S phase of interphase, so I have a second identical chromosome right next to it. This chromosome 1 came from my mom. It is maternal. Now, I'm a diploid individual, so I have 2 sets of chromosomes. I've got maternal DNA from my mom, and I've got paternal DNA from my dad. So I also have another set of chromosome number 1s. This one comes from my dad. In a normal skin cell, there's 1 copy of each. And I'm going to create haploid cells, so I need to duplicate everything. I've now got my maternal and my paternal DNA copied. So each one of these is called a sister chromatid. These 2 together, my maternal and paternal DNA, are called homologous chromosomes. Homologous chromosomes. Homologous means that they're the same type. It's chromosome number 1. I just got one from mom and one from dad. Now in prophase 1, this is where I'm going to take my maternal and my paternal DNA, and I'm going to kind of lay them on top of each other. They're going to smush real close together. So here's my chromosome number 1 from mom. There's my kinetochore. And then I'm going to take the chromosome number 1 from dad, and I'm just going to like lay it right on top. So I'm going to fill in chromosome number 1 from dad a little bit darker so that we can see that they're 3D, right? So chromosome number 1 from dad is sitting right on top of chromosome number 1 from mom, and that allows the maternal and paternal DNA to kind of smush together. So this is called the Synaptonemal Complex. It's a complex because it's more than one thing. Like, you may live in an apartment complex that has more than one apartment. So that's what complex means. And synapsis is what's occurring. The DNA from dad and the DNA from mom are like smushing together. And it's important because they're going to swap information. Where I had only information from mom on some of these chromosomes, I'm going to take a little bit of dad's and I'm going to smush it in. So that occurs in the Synaptonemal Complex in prophase 1. Now in prometaphase 1, I'm still in my Synaptonemal Complex. So there's my paternal DNA, and then right behind it, I've got my maternal DNA. So they're sitting together super tight still. And in prometaphase 1, I'm going to attach my microtubules at the kinetochore, which is going to help me move my homologous chromosomes into the center of the cell for Metaphase 1. My homologous chromosomes. Make sure you can still see that. So the homologous chromosomes line up in the center using the microtubules that I attached to my kinetochore. So I'm going to like pull this around and move it until they're all lined up. And then in Anaphase 1, anaphase for away, I'm going to use those microtubules and I'm going to pull apart my homologous chromosomes. Except where I once had all of the information from my maternal DNA in one place, during that Synaptonemal Complex, I've taken a little information from the paternal chromosomes. The paternal chromosomes have done the same thing. They've taken a little bit of information from the maternal chromosome number 1. That process of swapping information is called crossing over. Crossing over. And it occurs in prometaphase 1. So that's what's happening with my synaptonemal complex, is I'm allowing crossing over to occur so that when I pull my two homologous chromosomes apart in anaphase 1, they're no longer genetically identical. They're now recombinant. Oop, I think there's an extra in. Recombinant. So super exciting, because my goal here is to make non-identical cells. And if I didn't do crossing over, I'd be left with identical cells. So I want to take the information from mom and dad and swap them. And that's what happens in crossing over. And then I pull them apart in anaphase 1. In telophase 1, you're going to pretend like it's normal in mitosis. So the chromosomes will kind of go to the edges and you'll start to pinch your cell in so that you can do cytokinesis. In animal cells, cytokinesis is occurred with a cleavage furrow. Super weird name, right? Do you want to know why? Because of this. So this is my computer screen. I am showing you a cell that is undergoing either mitosis or Meiosis One. And what happens is you wrap a protein around the outside of your cell and you start to cinch it tight, kind of like you're pulling a lasso tight. And as you do, it contracts and contracts and contracts, and it kind of cuts your cell into two. And as it does, it looks like a butt or cleavage. And so it's called a cleavage furrow because you're literally cleaving one cell into two cells. So we did crossing over in prophase 1, and we pulled our chromosomes apart in anaphase 1, and in telophase 1 we got ready to do cytokinesis for the first time. Now, all of this is to make recombinant DNA, which is just DNA that's not the same as what you started with. And there's actually one other thing that happens in metaphase 1 that's important for making non-genetically identical cells, and it's called independent assortment. Okay, I'm abbreviating it so I don't run out of room. Independent assortment occurs in metaphase 1, and let me show you how this works. Okay, here's my computer screen again, and this is showing us independent assortment. So independent assortment refers to how your homologous chromosomes line up on the metaphase plate during Meiosis 1. And this only happens during Meiosis 1 because in anaphase 1, you pull your homologous chromosomes apart and you no longer have homologous pairs. You're left with just sister chromatids. So here on the right, we have the paternal DNA and the maternal DNA on opposite sides, so that when my cell does the first cytokinesis and I'm left with just my sister chromatids in each of the cells that result, one of them has just paternal DNA and one of them has just maternal DNA. I'm going to do a second round of cytokinesis because I want to end with 4 cells. This is my goal. I'm ending up with 4 cells. And 2 of those cells are maternal and 2 of them are paternal. So that's one option. But independent assortment means that whoever's on the left and whoever's on the right, it doesn't really matter. So if the chromosome number 1 from dad's on the left, you've got a 50-50% chance that chromosome number 2 from dad will be on the left or the right. They're independent of each other. Their assortment does not rely on the other chromosomes. So now when I do my first cytokinesis event and I end up with 2 cells, this one's not only maternal DNA, and this one's not only paternal DNA. So it gives us more results. I do my second round of cytokinesis, and now I have 2 cells that have chromosome number 1 from dad, chromosome number 2 from mom, and 2 cells that have chromosome number 1 from mom, chromosome number 2 from dad. And that means that of these 2 chromosomes, I have 4 possible variations that can occur. It just increases the diversity. All right, so now we just did cytokinesis the first time, which means we achieved it with a cleavage furrow. You saw that. After the first cytokinesis event, so after cytokinesis 1, there's a couple things to note. You now officially have 2 haploid cells. They're haploid because you separated your homologous pairs. Homologous pairs are 1 copy from mom, 1 copy from dad, which means you have 2 copies. You have 2 sets of chromosomes. You're left now with just 1 set of chromosomes. Ran out of room. That says chromosomes. After your first cytokinesis, you still have sister chromatids. My sister chromatids are still attached, which means that I still have double the DNA. So I need to fix that. I don't want to have sperm and egg that have double the sets of chromosomes, double the number of chromosomes, excuse me. So to fix that, I'm going to do a second meiosis event, and it's going to follow the same steps as mitosis, and it's going to look really similar to mitosis because all the really special things that happened in meiosis already occurred. So in prophase, if the chromosomes started to decondense, they'll recondense. If the nuclear envelope was forming, it will deform. It'll disappear again. In prometaphase 2, the microtubules will attach to the kinetochore, but this is only happening on 1 set of sister chromatids. So no more Synaptonemal Complex, that already occurred. In metaphase, they line up in the center, and then in anaphase, I'm going to take my 2 sister chromatids and I'm going to pull them apart. Each of them will go toward the edges in telophase, allowing cytokinesis to split them completely into different cells, and that will leave me with 4 total cells where I started with 1. And those cells are going to not be genetically identical. They're not genetically identical because when I started with my DNA, I did crossing over during prophase 1, which allowed me to swap some DNA from my paternal chromosome and put it on my maternal chromosome. Basically, I recombined my DNA, it's recombinant DNA. Then I aligned them randomly on the metaphase plate in independent assortment that occurs during metaphase 1. And when I did anaphase 1, I pulled apart my homologous pairs. So I pulled apart my homologous pairs in Meiosis 1. Then, in Meiosis 2, everybody lined up on the metaphase plate, and this time when I pulled them apart, I pulled apart my sister chromatids. So anaphase, you pull apart sister chromatids. Then I do telophase and cytokinesis 2, and at the end, so at the end of meiosis, I have produced 4 cells. They are not identical, and we call them germline cells. Germline because they're going to become sperm and egg. Alright, and so that's how meiosis occurs, and that's what this plant has been doing to produce eggs up in the base of these flowers, and to produce all this fabulous pollen that got everywhere until I pulled off the pollen sacs. So, you're doing meiosis, this plant's doing meiosis, anything that does sexual reproduction versus asexual reproduction will use meiosis, including fungi, which don't use sperm and egg, but use spores, which is a whole different story and is really, really exciting. So, thank you for watching the video. I hope you have a lovely rest of your day. Email me with questions and I will talk to you soon.